So in what situation would you actually want to transcribe the lac operon? Well, we just said that there's kind of two things that the cell needs to be measuring. The first thing is whether or not there is lactose around. But the second thing that's even more crucially important is whether there's glucose. If there's glucose around, this cell does not want to turn on the lac operon because it prefers to use glucose. So how does the cell sense when glucose is around? We just looked at how it senses when lactose is around. So this is part of the positive control of the lac operon, and this involves a third site. So now we've talked about the operator sequence, the promoter sequence, and then we mentioned that there's this activator binding sequence. This was one of the third cis-acting elements that we described. There is a site here which can bind to an activator protein. And this activator protein gives us the most robust transcription. So let's take a look at how all of this works and kind of walk through this schematic. Let's consider the case where there is high glucose, but there's also high lactose. So in the case of high lactose, we know that the repressor will be bound to the inducer, which is lactose, and it will come off of the operator. So the RNA polymerase potentially can bind and transcribe the LAC-Z, Y, and A genes. However, because there's high glucose, we know that the cell actually doesn't really want to make these enzymes. It prefers to just use the glucose. So it turns out that even though the RNA polymerase can bind to the promoter, it does not bind well. Remember when we looked at -35 and -10 sequences, depending on how closely those sequences match to the consensus, will determine how robustly the RNA polymerase and sigma factor will find that particular promoter. So in this case, RNA polymerase does not find the promoter very well without additional binding. So now let's consider a second case. This is the case where lactose is high, so the inducer binds to the repressor and pulls it off of the operator sequence. However, glucose levels are low. Okay, so in this case, this is actually a time when we would want to transcribe the lac operon. In this case, low glucose levels, an activator protein, which is called CAP, can actually bind to the activator binding site, which is called the CAP site. And this helps to keep RNA polymerase tightly bound to the promoter. This results in frequent transcription, which means that you get a lot of LAC-Z, Y, and A produced. So even in the presence of lactose, the lac promoter can be weak. And the question is, why does this happen? That can happen whenever glucose levels are high. So the RNA polymerase does not bind optimally to the promoter sequence. It's only when glucose levels are low that the CAP protein can essentially hold the RNA polymerase tightly bound to the promoter so that you get frequent transcription. So the lac operon will only be turned on when lactose is high and when the glucose levels are low, so that you get this CAP binding to the CAP site. So this is a second form of regulation. This is positive regulation. So the lac operon is positively regulated by a protein called catabolate activating protein, or CAP, which only binds in the absence of glucose. So now we have two different forms of regulation. We discussed the negative regulation by the repressor binding to the operator sequence and decreasing transcription of the operon. Now we've discussed a second form of regulation that is actually positive regulation. And so this is the mechanism that's mediated by this CAP protein binding to the CAP binding site and increasing or stimulating transcription by RNA polymerase. So how exactly does the cap binding stimulate transcription? And how does this cap sense the glucose levels in the cell? It turns out that the cap protein can only bind to the cap site when it is bound to another component, cyclic AMP. And we've discussed this cyclic AMP quite a bit when we talked about cell signaling. The cap protein will only bind to the sequence when it has cyclic AMP. And in this case, once it binds, it can enhance the binding of the RNA polymerase. So how does this work to read out the levels of glucose in the cell? How can cap sense glucose levels? It turns out that, as I mentioned, it will only bind to this cap protein will only bind to the DNA when it is bound by cyclic AMP. And cyclic AMP serves as a cellular readout for glucose concentration. And these two are inversely related. So in high glucose levels, there will also be low cyclic AMP levels in the cell. In low glucose levels, there will be a higher cyclic AMP concentration. So you can see here that if there was high glucose, there would be very little cyclic AMP, which would mean that there would be very little cyclic AMP cap complex that would be available to bind to the activator site. If, however, the cell was low on glucose, there would be high cyclic AMP levels, and this would allow the cap and the cyclic AMP to form a complex and bind to the cap site to activate the RNA polymerase binding to the promoter. So for those of you who like a little bit more explanation of this, the reason that there are low cyclic AMP levels when there's high glucose levels is that there's a relationship between glucose transport and the levels of cyclic AMP. So it turns out that when glucose is transported into the cell, it actually inhibits adenylate cyclase. So if there's high glucose, adenylate cyclase is inhibited, which can lower the cyclic AMP levels. I don't really expect you all to know this, but this is kind of for those of you who do like the extra explanation of how cyclic AMP can be a readout for glucose. What I do want you to be able to understand is that when we have high glucose concentrations in the cell, this will correspond to lower cyclic AMP levels and vice versa. So this is the level that I do expect you to understand this relationship. Okay, so what I would like you to do is to take a few minutes to draw the components of the lac operon. So you'll want to sketch out what are the structural genes, what are the regulatory sequences, and I want you to indicate whether the lac operon would be expressed or turned off in each of these different environmental conditions. So you can draw the lac operon itself and also draw which of the transacting factors would be bound in each of these cases. So A is the absence of lactose and the presence of glucose. B, presence of lactose, presence of glucose. C is presence of lactose, absence of glucose. And D, absence of lactose and absence of glucose. So what would that look like? You can pause your recording now to try to draw this out, and I really encourage you to do so. Okay, so I mentioned that the scientists that essentially worked out the details of the lac operon were doing so in an era when there was no DNA sequencing, there was no biotechnology that had been developed. So how did these scientists actually figure out all these details without doing any DNA sequencing? Turns out that Jacob and Monod were using classical genetic approaches to determine, which parts of this operon were cis-acting elements, trans-acting elements, etc. So the way that they did this is that they isolated mutants that had mutations in somewhere in the lactose metabolism system. And they used these mutants to kind of dissect the lac operon itself. This is a classical genetic approach, and this is actually the way that many scientists have gone about studying various different systems in the cell. So to carry out a forward genetic screen like this, the first step is to mutagenize your bacteria. So you want to induce a whole bunch of mutations. And the way that they did this, or the way that you can do this, you can use X-rays, you can use UV light, you can use various DNA damage chemicals. And you'll read about a number of different DNA damage chemicals that are often used in genetic screens. The most common, I think, that you hear about are EMS, ethyl methane sulfonate, which is just used to induce a whole bunch of mutations. And these mutations, of course, are going to be random, so you have no idea which genes you'll be hitting when you randomly mutagenize your bacteria. The second part of this experimental design is to design some kind of a screen to identify mutants of a particular pathway. So in this case, Jacob and Monod were interested in looking for mutants that were unable to metabolize lactose like normal. So how could they screen the mutants? How are they going to identify these mutants that have particular mutations in the lactose pathway? The way that they did this is through replica plating. So they essentially had a master plate that was grown in the presence of glucose. And they then wanted to test, so they knew that all of these colonies, each colony represented a different mutant that had a mutation in a different gene. They then wanted to test whether these same mutants could grow if lactose was present. Could these actually metabolize just lactose in the absence of glucose? So they transferred these colonies to a velvet covered block, and then they replica plated them to a new plate which only had lactose present in the medium. And so then they were looking for colonies that grew okay on the glucose plate, but now were unable to metabolize lactose. So these would, of course, be their mutants in the lactose metabolism pathway. So through this kind of analysis, they did a number of different tests here, but through this kind of analysis, they were able to identify two general sets of mutants. They found some mutants that were constitutively expressing the lac operon. And what that means is that they turned this thing on no matter whether there was lactose around, no lactose around, glucose around, no glucose around. These mutants were turning on the LAC-C and LAC-Y gene all the time. They also found a set of mutants which they called uninducible. These were mutants that were unable to turn on the lac operon even if there was an inducer around. Somehow, these mutants could not respond when there was an inducer around. So these were the two major classes of lactose metabolism mutants. And these different mutants allowed them, Jacob and Monod, to essentially dissect all of the different components here and to figure out which elements were binding to which sites, etc.